Okay, so let us continue. In my first class, I think I have discussed the basics of cell signaling and their significance in multicellular and unicellular individuals. Also, I described the types of signaling as well as the principles of signaling, the mechanism, the brief mechanism, the what is receptor and what are the basic mechanisms of receptor, and what a receptor does uh, in case of signal transduction, the properties of signal transduction, classification of receptors. Okay, now let us go further into details. Now, as I have said earlier, that different kinds of receptors are present in different location of a cell depending upon the nature of the ligand, the basically the solubility of the ligand. So depending upon solubility of the ligand, the receptors can be extracellular for those ligands, as far as, if you remember, those ligands which are easily soluble in water or hydrophilic solutes or ligands or signals, the receptor will be present at the surface or cell surface receptors. Those molecules which are not water soluble, rather non-polar molecules. So the polar molecules would have basically an extracellular receptors. The non-polar are hydrophobic molecules or lipophilic molecules or non-polar molecules. They would have basically an intracellular receptor, either cytosolic or nucleus. So there are examples of different kinds of cytosolic receptors. As far as I can remember, most of the steroids have cytosolic receptors, but an unusual nuclear receptor is present for the hormone which is known as thyroxine. As we all know, the thyroid hormone, that thyroid hormone is basically works by getting inside the target cells and then goes inside the nucleus, and the receptor, which is basically a transcription factor, converts, which was in absence of the ligand, which was a repressor. In presence of the ligand, as the ligand binds to the receptor, it becomes a transcription activator and basically gives or elicits a genetic response. Now, so these are the different kinds of signaling molecules, starting from hormones to different kinds of hormones, the tropic hormones, the different kinds of other small hormones, the catecholamines, the protein hormones, the steroid hormones, vitamins also, the derivatives, different derivatives of other molecules, some xenobiotics, means some foreign chemicals which are known as drugs, which are basically that we consume to fight different kinds of pathophysiological conditions or diseases. Even the cytokines, growth factors, these are all ligands, and they have a different, there are whole array of molecules starting from protein to lipids to glycolipids to glycoproteins, even gases, carbon monoxide and nitric oxide. So these are different kinds of ligands that we have got. Now a basic question comes when a ligand or a signal molecule, suppose acetylcholine, elicits differential responses in different kinds of cells, as we are seeing right now. In the cardiomyocytes or heart muscle cells, it decreases the rate and force of contraction of the cardiomyocytes, whereas in skeletal muscles it induces contraction, and in case of salivary gland cells it stimulates excretion of salivary glands. That means to secretion, it exits or it induces secretion. Now how a single molecule can elicit differential responses in tissue specific manner, that is the basic question, right? So to understand this, we have to go just a little back, as I have discussed that the mechanism, that the main components used in signal transduction are receptors and downstream molecules. So, the path, the response that we get is entirely dependent upon which receptor is activated, and following the receptor activation which downstream molecules are activated. Okay, to know, therefore, the components of signaling are basically receptors and the downstream signaling molecules. Since tissue specific gene expression occurs, therefore, the cardiomyocytes, skeletal muscles and salivary glands all have differential protein expressions. Therefore, it is likely that the downstream molecules of all of these cells are different. Additionally, some cells such as cardiomyocytes and skeletal muscles, they have different kinds of receptors. See? So, since they have different kinds of receptor, the same molecule activates two different receptors, therefore, it elicits two different responses or tissue specific responses. However, the cardiomyocytes and the salivary gland cells, they have similar receptor, but yet they are giving different kinds of responses. One, it is decreasing the rate of contraction, one other is secretion, or it is basically inducing secretion. Therefore, since the receptor components are same, therefore, the main changes in these cells are mainly the downstream molecules. So, now we get to the question that if somebody asks in a competitive examination that a single ligand, why a single ligand elicit differential responses in tissue specific manner. So, I guess you should answer it in this specific manner that suppose the main components are receptor and downstream molecules. So, the receptors may be different or the downstream molecules may be different, therefore, they elicit different kinds of response. So, this is, I think, how you should approach your questions during your higher studies or your comparative studies. Now, so regarding response, so since after induction of or after activation of the receptor, there are different kinds of cellular responses, okay, depending on which proteins or which downstream molecules are activated. But depending upon the nature of cellular response, okay, there are broadly divided into two different or two distinct kind of responses. One which is known as physiologic response, another is known as genomic response. So, these two responses are basically different regarding the mechanistics or the entire kind of response that a cell is evoking as the signal binds to the receptor. Now, what are these two kinds of responses? Now, let us look at in these two responses. Now, after the signal binds to the receptor, the physiologic response is basically elicited very fast, and this fast physiologic response is elicited by altered protein function. That means, as I have told you earlier, that there are some downstream molecules which are known as effector proteins, or which proteins are basically enzymes or basically which directly functions. Understood? So, they may be some metabolic enzymes or maybe some cytoskeletal proteins or maybe something else, but whatever they may are, they give the response directly. So, the in physiologic response such as change in cell shape or change in contraction rate, such as let us take the example of salivary gland secretion, or let us take the example of this decreased rate of contraction. All these are elicited by, all these are basically very immediate responses such as this one or this one, and they are elicited mainly by changing the protein functionality, and this change in protein functionality basically occurs by post translational modification, either by phosphorylation or by dephosphorylation or by altering different kinds of molecular switches, which elicits direct change in cellular behavior, either by opening of a ion channel or maybe change in cell shape or maybe secreting something or maybe taking up something, all these different kinds of, which are very fast and which are instantaneous response. Another kind of response, as we all know, this is genomic response. This kind of response occurs in the level of the gene. So therefore, that means it alters the transcriptional profile. After the receptor binds, the receptor activates some downstream molecules that ultimately changes the transcriptional status or transcriptional nature of the gene of the cell. That means it basically accelerates transcription, or basically deaccelerates transcription, or retards transcription of some genes, or accelerate transcription of some genes, and therefore the new protein synthesis, the rate of protein synthesis, that means the rate of the transcriptional status of the cell gets changed, and therefore it gives a very long lasting response. Okay, and this kind of response is basically very slow, as you can say. Let us take the example of hormone thyroid. So thyroid, as we all know, it has a nuclear receptor, it elicits a genomic response, and the response is a very widespread or very long lasting response rate. Whether the physiologic response is very instantaneous, let us take the example of adrenaline, so which gives a very instantaneous response, but genomic response takes quite a while to set in, and therefore takes quite a while to let go also. Okay. Now how these responses basically are instantaneous or very fast, since there are a lot of responses which needs to be very fast. In order to respond accordingly, the response needs to be very instantaneous or fast to respond accordingly. Suppose seeing a snake or a tiger, or suppose seeing a predator, so or seeing a prey, so the individual have to give a very immediate response, or else the entire game will be lost. So therefore there should be some direct, so the signaling should act like direct some switches, just like electricity. Suppose when we sweat a lot, or suppose when we feel a lot of heat, we turn on our fan, okay, and by turning on the switch, immediately the fan starts rotating and we feel comfortable, and after a bit we just if we feel if the need of the fan, or if the need of that air, cool air, is not anymore, so then we switch it off, right. So there's the main mechanism is that switch is very instantaneous, it elicits its effect instantaneously. Now in order to give rise to a instantaneous response, there should be some molecular switches, so some proteins which can be very instantaneously turned on and very instantaneously turned off, right, and these kind of switches are known as molecular switches. So if a signal comes, it becomes from off to on state, and if signals goes out, it becomes from off on to off state, understood, instantaneously. One of such there are two such basically molecular switches which elicits these kind of responses. One of them is being phosphorylation. So phosphorylating a protein basically changes its conformation and therefore changes the activity, and this kind of proteins phosphorylation switches is basically initiated by protein kinases that basically phosphorylates a protein by adding one phosphate group from ATP. The ATP is converted into ADP, and the released phosphate is added to the protein. So therefore the protein's conformation changes and it becomes active. So the protein kinases are basically activators of phosphorylation, and there are also another kind of proteins which is known as phosphatases. As the name suggests, it removes the phosphates and therefore it again goes to inactive conformation. Similarly, there is another kind of switch which is known as GTP switch or GTP proteins. Okay, these are very widespread molecular switches, and the uniqueness of GTP binding proteins, they are, or you can say G proteins, there are monomeric G proteins and trimeric G proteins. Here there is the monomeric G proteins. The example is of monomeric G proteins. The monomeric G proteins becomes on whenever it binds to a GTP. So, if signal comes and GTP binds to the these G proteins, they become active. When this attached GTP, since these molecules they have also inherent GTPase activity, and if these molecules they hydrolyze their GTP from GTP to GDP, therefore when it hydrolyzes the GTP, it becomes the GTP is converted into GDP and the molecular switch becomes inactive, and therefore the activators of these are the active conformation is GTP bound state and inactive conformation is GDP bound state, and this GTP bound state or active conformation is elicited by GTP binding, therefore it is known as guanine nucleotide exchanging factor. So, GEF, these are the molecules that activate them, and there are some molecules which inactivate them, which are known as GAP or GAP or GTPase activating proteins. So, there are some proteins which activates the GTPase activity of this G protein, and when these GTPase proteins activate this GTPase activity, the protein from GTP becomes GDP and it hydrolyzes the GTP and it becomes inactive. So, these are the main molecular switches that activate. Okay, now, so a basic question of genomic response comes that a question may come that why or how genomic response can be amplified. Since we have all learned about the different kinds of molecules, there is an amplifier molecules in the cascade mechanisms which basically amplifies the response, but also there is a question since the extracellular receptors or which gives physiological response, they have lot of molecules which can amplify themselves, these proteins, and therefore it can elicit a amplification or amplified response. But in case of genomic responses, since the receptors basically are transcriptional activators and they activate when the receptor becomes active, it basically gives rise to some proteins, and these proteins again in the so at the primary response, the receptor bound hormone or the hormone bound receptor becomes active and it transcribes a lot of protein, okay, or few proteins from some responsive elements or responsive genes or their promoters. But when this primary response is elicited, the primary response proteins, they are inherently are transcriptional activators for other secondary response genes. So, therefore, this kind of second level or two levels or two stratas of responses basically amplifies the extent of genomic response and therefore gives a very long lasting effect. So, this is how the long lasting effect of genomic response occurs by basically by primary, secondary, and also there are some hormones which also elicits a tertiary response. Understood? Now, so now let us directly go or let us directly jump into G protein coupled receptors. So, and what are the basic mechanisms? So, there are some receptors, as I have told you earlier, these proteins or these receptors are known as G protein coupled receptors, and these G protein coupled receptors, just a second. Okay, now these receptors are known as G protein coupled receptors because they mediate their action via a cell or via a protein which is known as a G protein. Okay, so that is why they are known as G protein coupled receptor or GPCR, their acronym. They are popularly known as GPCRs, however. And so this is the receptor which looks like a snake, right, and the G protein which is the mediator of this receptor mediated activity is this one, okay. and this is a trimeric protein composed of 3 kinds of polypeptide chain, alpha, beta and gamma, this G protein, and they are basically nothing but, as I told you, they are also some molecular switches. So they are nothing but the GTP binding switches, or they use these switches are turned on by binding of GTP, and these switches are turned off when these GTPs are converted into GTPs, right. Now, how do they elicit a particular response? So, since a G protein coupled receptor has a receptor, and G protein being very important downstream molecules, so therefore there will be other some downstream molecules and effector molecules that will elicit these kinds of response. So, let us first come into the receptor. So, the receptor is basically a snake like molecule present, and it has 3 domains. One is the extracellular domain, which is present outside the cell. There is an intracellular domain which is present inside the cell, and there are some part of domain which is known as transmembrane domain. This is present, or this is basically present inside the plasma membrane, and this single polypeptide chain crosses the plasma membrane 7 times. Okay. So, that is for they are also known as 7 pass transmembrane receptors, right. Now, as the receptor, as the ligand, so the receptor is basically is far away from the GPCR, or in some cases they are also attached with the G protein. The receptor is also attached with the G protein, or in some cases they are quite far from the G protein. However, whatever it may be, the cytosolic phase of the receptor is very near to the cytosolic or the G protein, which is in the very near to the cytosolic phase of the cytosolic phase of the receptor, this G protein is. Now, what happens as the signal molecule binds to the receptor, the conformational, there is a conformational change in the receptor. As we all know, there is the signal and receptors, they undergo a non-covalent interaction and reversible interaction, and this kind of non-covalent reversible reaction basically changes the conformation, since the binding of such biomolecules with another molecule, that is the receptor, changes the non-covalent interactions. That means it may be the hydrogen bondings, or the van der Waals force, or the hydrophobic interaction, or ionic interaction. Some of these components, they alter the folding pattern of the protein, and therefore it basically the folding pattern of the receptor change in such a manner that this protein, or this receptor, activated receptor or ligand bound receptor, acts as a guanine nucleotide exchanging factor, or GEF. That means when at this particular conformation, when the receptor becomes in or comes to this conformation, it binds to that G protein, okay, and it elicits the alpha subunit to release its bound GTP and to take up a new GTP. Okay, so it induces the alpha GTP or alpha subunit to release its GTP and to take up GTP, and as it takes up GTP, the alpha subunit takes up a new GTP and releases the GTP, and the alpha GTP, there is a conformational change of the alpha GTP, and the alpha GTP becomes active and it dissociates from the remainder beta gamma complex. Clear? So, this so I should tell you firstly that what is this G protein. This G protein is composed of 3 polypeptide chains, alpha, beta and gamma, and this alpha is basically present. The alpha can be present in 2 states, either it can be found in GDP bound state, or it can be found in GTP bound state, and this alpha is basically nothing but it can be analogous to the monomeric GTPases, okay, or monomeric G proteins, and this alpha has the inherent GTPase activity. So, and this beta gamma has some other factors or some other functions. So, in this trimeric G protein what happens when the receptor binds to the alpha GTP, the alpha sorry alpha GDP, the alpha subunit release its GDP, takes up GTP and it becomes active and it dissociates from the beta gamma complex. So, this is how the mediator of G protein coupled receptor is activated. Okay. Now, I think till this part everything is normal, and there are, depending upon what kind of substrates that this alpha GTP subunit activates, G proteins can be further classified into two types. One is known as GS, okay, another is known as GQ. Okay. Now, GS basically activates adenylate cyclase and GQ activates phospholipase C gamma pathway. Okay. Now, so we are first going to talk about GS. Now, the ligands of these GSs can be all these ligands, as they are mentioned, different kinds of tropic hormones and synthetic TSH, leucine, adrenaline, parathormone, different kinds of hormones. There are a lot of kinds of hormones that basically activates this GS. That means they basically activate the alpha subunit of GS, and this, now the GS alpha subunit activates another transmembrane protein or integral protein which is known as adenylyl cyclase, okay, which is also its catalytic subunit is also present near the cytosolic region, okay, and it has an integral transmembrane part. So, as the alpha GTP or the alpha activated alpha GTP binds to adenylyl cyclase, this causes two-way functions. First, as adenylyl cyclase, as the alpha GTP binds to adenylyl cyclase, the alpha subunit's GTPase activity becomes activated. So, therefore, it hydrolyzes the GTP into GTP and itself becomes inactive and again go and binds to the beta gamma subunit. Understood? So, this is how so we have shown how the activation occurs. The activation occurs by the receptor activity which acts as a GEF, and the adenylyl cyclase which acts as a GAP, that is GTPase activating protein, that hydrolyzes or that induces the hydrolysis of alpha GTP's hydrolysis, and it again inactivates alpha subunit. But as the adenylyl cyclase inactivates alpha subunit, it relays or it activates some other relay proteins, and adenylyl cyclase being an enzyme itself, it basically converts a lot of molecules. It is a main amplifier which produces a lot of second messengers, as we all know, and that second messenger is known as cyclic AMP. That means it has a cyclic phosphate or phosphate bond within that adenine nucleotide. So, it converts ATP or adenosine triphosphate into cyclic AMP or adenosine monophosphate. Understood? And this enzyme creates a high number of molecules of this cAMP, and this cAMP then now can diffuse from one region to another region. Since the signal molecules is known as the first messenger that induces the signal transduction through receptors, the second messenger basically amplifies the number of signaling molecules, further downstream signaling molecules, and it amplifies the localized activation of receptor from the localized or local cellular or local subcellular activation of some proteins till the entire cell becomes active and the entire cell's machinery becomes activated. So, this is why they are known as the second messenger. So, after cAMP binds or cAMP is produced, cAMP act as a second messenger and it activates a protein kinase, a serine threonine kinase. That means a kinase means which basically phosphorylates other proteins, some amino acid residues, and serine threonine kinases are those enzymes, they phosphorylate the serine or threonine amino acid of their target substrates. Okay. So, previously basically the PK is composed of 4 subunits. 2 of them are catalytic and 2 of them are regulatory. Since we all know that different kinds of enzymes are known are there, this kind of enzyme is known as a allosteric enzymes. That means apart from presence of a substrate, it should need another some factors which are known as regulators, which and if those regulators are present, only then these enzymes will elicit their function, else in presence of the substrate also these, and in absence of the regulators, these proteins or these allosteric enzymes won't elicit their activity out. So, this is pKa is one example of allosteric enzyme. Now, as cyclic AMP binds to the cyclic AMP acts as the regulator and binds to the inhibitory subunit or regulatory subunit. This regulatory subunit was basically inhibiting the catalytic subunit previously. As the second messenger or cAMP binds to this regulator, there is a conformational change of the regulatory subunit and the regulatory subunit basically falls off. Now, as these inhibitors of the catalytic subunits are off or distant away, the activated or the catalytic subunit become activated. So, this is how the cyclic AMP or their regulator binding activates the regulatory or the allosteric enzymes. Now, after activation, this pKa, however, this pKa, the name suggests protein kinase A. I think you have known already why they are known as protein kinases, but A stands for cAMP. That means, since their activation is dependent upon cAMP or adenosine monophosphate, they are known as pKa. There are other kinds of enzymes which are known as pKc also, and their activation is dependent upon calcium. That is why they are known as pKc. C stands for calcium. Now, here this is pKa. So, GS activates adenylene cyclase, adenylene cyclase activates pKa. Now, pKa being the effector enzyme, it elicits different kinds of physiological responses by activating a lot of different other metabolic enzymes or different other kinds of ion channels or different other kinds of cellular cytoskeletal. So, to give rise to a physiological response. However, it can also elicit genomic response by it itself goes inside nucleus, it activates or it phosphorylates a particular protein, and this protein is known as CREB, that means cAMP responsive element binding protein. That means this CREB